guys, in the last video we were done with the processes of translation in which protein were made by the help of RNA. Alright, so today we are going to discuss about the DNA replication. DNA replication, as you can see the topic here. So what will be happening in DNA replication? Alright, in this process DNA can make its copy as well. DNA will be made from DNA itself. Alright. So basically this process is known as DNA replication. Now we need to know that how many types of DNA we do have. Let's see here. Types of DNA. Alright. So first type we do have, that is linear DNA. Linear DNA. Alright. This is the linear one. Like this. Here we do have DNA. Here are the nucleotides of DNA. Alright. These are the nucleotides. These are the nucleotides. And other type of DNA we do have, that is circular DNA. Circular DNA. Alright. So here I'm just going to make it now. This is the circular DNA. This is the cell basically. Here we have circular DNA. Alright. This is the circular DNA we do have. So we are having two types of DNA. The first one is linear DNA. Linear DNA. We do have linear DNA. All humans have linear DNA. While this is the circular DNA and bacteria have circular DNA. Alright, these were the types of DNA. Alright, now we are going to see if DNA can make copy of itself. So all cells of humans having same DNA. Let's see. Let me scroll down. For example, if this is the cell of skin in human case, alright, in human case this is the nucleus, and we know that replication will be happening in the component of cell, which is the nucleus. Alright, it will be happening in the nucleus. So basically this is the DNA we do have, and this is the skin cell. Alright, this is the cell of skin, and now I'm just going to make the cell of pancreas. Alright, this is the cell of pancreas. This is nucleus, and here we do have DNA. Alright, this is pancreas. This we have pancreas cell. Alright, so here we know that here is the insulin gene. Alright, here we have insulin gene. Let me write here. Insulin gene. But now here we need to focus on one thing. If here we have insulin gene and we know that this DNA is replicated. This DNA is replicated. Like this DNA is made from this DNA. Alright. So here we do have insulin gene as well. Alright. If in pancreas cell we do have insulin gene, so means in the cell of skin we do have insulin gene as well. Because this is the replicated DNA. Alright. So here we have insulin gene as well. So now we need to know that insulin gene is secreting, like it is secreting its secretion in the pancreas cell. But why it's not secreting its secretion in the skin cell? Just because of the activation. Here the insulin gene is activated. It is activated. Alright. This is activated. While talking about this insulin gene in the skin cell, that is inactivated. Just because of this reason we do have inactivation of insulin gene in the skin and activation of insulin gene in the pancreas. Just because of this activation we do have different secretions in the different cells. Alright, so here insulin gene is activated in the here insulin gene is inactivated in the skin cell. Alright. So now we are going to discuss about the DNA replication, how DNA is replicated. Alright, so for suppose this is a DNA I have, wait, let me make it straight. Okay, so here I have one strand of DNA. Alright, I'm just going to select another, and here I have another strand of DNA. Alright, here are the nucleotides. These are the nucleotides. As we are discussing about the DNA replication in the human, but talking about bacterial replication of DNA, so that is the same. While transcription and translation were the different processes, those weren't same in the bacteria case and in the human case. While talking about replication of DNA, it is same in both. Just there we do have linear DNA and they do have circular DNA. Alright, so this is the linear DNA we do have. So now what will be happening? Like these both strands will get separated, because we will not make like this will form here this strand and then this strand will form this strand. So they both will be new strands and they will be complementary to each other. But we don't have to do like this. We have to separate these both strands first. So let's separate it first. This I have one DNA strand, and here I have separated this as well. So here I have another DNA strand. Alright. So now wait, let me make it with the new color. Okay. So now what I'll do, this strand will make copy of itself. So here will be new strand of this. Alright. And this strand will make copy of itself as well. So this will be new strand I'll do have. Alright. So here an enzyme will come and it will start putting nucleotides, complementary nucleotides. Alright. Nucleotides will be here. This will be the same process as well there too. So this will be the same. Nucleotides would be here, 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 and here. So this will be the same. Alright. So like this DNA will be replicated. Alright. So this type of replication is known as, wait, let me write here, semi-conservative replication. This type of replication is known as semi-conservative replication. Alright, because here in this case both all the strands are separated and then new strands are formed. Alright, so that type of replication is known as semi-conservative replication. Alright, so now we are going with the process of replication. Alright, let me scroll down. Okay, here it is fine. Okay. So basically what will happen? Let's see. Let me select it. Alright. So if I do have this DNA, alright. If I do have this another strand of DNA, these both are the all strands. Alright. Here I have the nucleotides of this. These are the nucleotides. Nucleotides. These are the nucleotides. These all are the nucleotides I do have. Here I have complementary nucleotides. Alright. These are the complementary nucleotides. Now let's see. Here I have G. Here I have C. Here I have T. Here I have A. Here I have C. Here will be complementary nucleotides of this. So here I have G. So here it will be C. Here I have C. So here it would be G. Here I have T. So here it would be A. If I have A here, so here it would be T. C will be complementary to G. So here an enzyme will come, which is the DNA A enzyme. Alright, this is the DNA A enzyme. So here it will come as well. It will break the bond between A and T, because we know that between A and T we do have double bond. Alright, we do have double bonds between adenine and thymine. While talking about cytosine and guanine, we have triple bonds between them. Alright, so double bond can be broken easily as compared to the cytosine and guanine because they have triple bond. So DNA A enzyme will come and it will start breaking the bond between A and T. As you can see here, A and T. It will start breaking the bonds between them because they do have double bond between them. So they'll be separated like this. Wait. After got separation, what will happen? It will make replication bubble. Alright, this is the replication bubble. Here is the replication bubble will be made. Alright, let me make nucleotides here now. These will be the nucleotides. Alright. Here will be nucleotides. Alright. Complementary nucleotides of this. Wait, let me make it here. Alright. So these will be the complementary nucleotides. Alright. Now it has still the power to join, to attracts to the complementary strand. So now what we have to do, we'll put here the proteins. What those proteins are called, basically those are SSB protein, which are single strand binding proteins. So here I'm just going to put these are SSB, single strand binding protein, so that it cannot attract towards its complementary strand. Alright, here I'm just going to put what these are, basically single strand binding protein, and this is also known as SSB, single strand binding protein. Alright, let me mark here, single strand binding protein. Alright, so now it has got separated. Alright, now what we have to do, we have to just cut it from the middle. Alright, I have cut basically. So now what I have, let's see here. Now I have structure like this. Alright, here is one strand, here I do have another. So basically this is the replication fork. Alright, this is the replication fork I do have, because I have cut replication bubble from the mid. Alright, so by cutting the replication bubble from the mid, what I have gotten, actually I have gotten replication fork. Alright, now I have the complementary nucleotides of this, here, and here as well. Then again complementary nucleotides on this strand. Alright, so now here an enzyme will come. What that enzyme will be, basically let me write here, DNA dependent DNA polymerase A. Alright, because we do have different types of this enzyme. So first of all, what we will be getting an enzyme which is DNA dependent DNA polymerase A. Why it is DNA dependent? Because this enzyme is dependent on the DNA. It will make itself copy. Alright, so that's why it is known as DNA dependent DNA polymerase. And A, because we do have different types of this enzyme, so that's why this is DNA dependent DNA polymerase. Now we do have some conditions of these DNA dependent DNA polymerase A enzyme. This is very efficient enzyme. This is highly efficient enzyme. That's why it has some conditions. Like it will always works on single strand, but from where it will start, it will work on double strand, and it will always make the strand from 5' to the 3' end. Alright, so here I'm just going to write here the characteristics. enzyme. Which enzyme? DNA dependent DNA polymerase enzyme. Alright, so we do have some characteristics of this enzyme, which is DNA dependent DNA polymerase. Alright, so I'm just going to write it down. Let me scroll down. Alright, so the first characteristic we do have of this enzyme is it always works on single strand. It always works on single strands. Alright, second characteristics of this enzyme we do have from where it starts, it always works on double strand. Alright, DS, which means double strand. Alright, third characteristic of this enzyme we do have, it always makes a strand from the 5' end and goes to the 3' end. That would be new strand or daughter strand. Alright, it always makes new strand or daughter strand from 5' end to the 3' end. Alright, these are some characteristics. The first characteristics we do have, which is it always works on the single strand. Alright, wait, let me mark here. It always works on single strand. As you can see here we made it single. This because of this enzyme, so that this enzyme can come and can work on this. Alright, so second characteristic we do have, from where it starts, it always works on the double strand. Alright, if it is going to start from here, then it has to be start from the double. Like here we have to put something to make it double. Alright, here we'll be doing that. No need to worry. Alright, so third characteristic we do have, it always makes new strand from the 5' end to the 3' end. Alright, if I have here 5' end and here I have 3' end, so mean the new strand which will be made, it will be made from the 5' end to the 3' end. If I am going to make the strand from here to there, so it must be 5' end, because this is the condition of this enzyme, 5' end, and here it has to be 3' end, so that it can be complementary to the template strand. Alright, so this was the second characteristic and that was the third characteristic. Alright, so now we are just going to check it out how this can be happen. Alright, now I'm just going to make the DNA replication strand again, the bigger replication strand, replication form. So basically I do have replication fork here. This is the one strand, this is an other strand I do have. Alright, so basically this is the 5' end of this strand, 5' end. If this is the 5' end, so it must be 3' end. This is the 3' end. And if this is the 5' end, because this is the complementary strand of this, so it must be 3' end. And here must be 5' end. Alright, this must be 5' end. Alright, let me make the nucleotides here. These are the nucleotides. Let me make it all. These are the nucleotides. Alright, here are the nucleotides as well, and here are the complementary nucleotides of this. These are the nucleotides, sorry. Okay, this is the nucleotide. Here I have nucleotides as well. So here are the complementary nucleotides. Alright, now DNA dependent DNA polymerase A will come and it start work just because of the conditions. Like according to the condition, it always works on the single strand. So this is a single strand and this is the single strands as well. Alright, second condition of that enzyme was from where it starts, it always works on the double strand. Alright, so here we have to put something to make it double. Alright, here I have to put something to make it double so that DNA dependent DNA polymerase can come and start works. Alright, so let me write here an enzyme which is a primase enzyme. It will come and it will put here the piece of RNA. So RNA will work as the primer here. Alright, because primase enzyme came here and it has put the RNA piece here so that it can be double and DNA dependent DNA polymerase can start working on this. Alright, if it has put here a primer, so here should be primer as well so that DNA dependent DNA polymerase can work here too. Alright, and we know that DNA dependent DNA polymerase will always make the strand from 5' to the 3' end. Like if this is the 3' end, we do have, if this is 3' end, so here must be 5' end. So we have to put the primer here because we know that DNA dependent DNA polymerase will always make the strand from 5' end to the 3' end. Alright, so now we have put the primers. Alright, these primers are the pieces of RNA. Alright, so now what will happen? Now DNA dependent DNA polymerase will start work. Alright, it will start make strand because now the conditions are fulfilled. Alright, here it would be made. Alright, this is the 5' end because we know that DNA dependent DNA polymerase always makes the strand from 5' end to the 3' end. So this must be 3' end. Here it would be 5' end. Alright, and here it must be 3' end. 3' end. Alright, in the first stand, like in the upper stand, we don't have to put primers again and again because this would be the continuous stand. Alright, but talking about the lower stand, so we have to put primers over and again. Alright, so here we have to put another primer so that it can be made. Alright, so here again it would be made. Alright, so this must be then 5' and then it must be again 3' end. Alright, so now what will happen? Alright, now this stand has some fragments. Alright, this is the one fragment, again this is another fragment. So these fragments are called what? Wait, these fragments are called what? Let me write here. These fragments are called Okazaki fragments. These fragments are called Okazaki fragments. Let me mark here. Okazaki fragments. Now an enzyme will come, like DNA dependent DNA polymerase A has worked now. Now we do have another enzyme that is DNA dependent DNA polymerase. Alright, DNA dependent DNA polymerase 3 will come now. Alright, now what it will do, it has some activities. Alright, first of all, it will always read template strand from 3' end to the 5' end. As this is an enzyme, let me mark here, DNA dependent DNA polymerase 3. Alright, it will always read the template strand from the 3' end to the 5' end. Alright, it will always read the strand from the 3' end to the 5' end. And second activity, it has exonuclease activity. What is that activity? Which means it will cut the nitrogen bases. Because DNA dependent DNA polymerase is highly efficient enzyme, it can put so many nucleotides in a second only. So alright, so here it has an exonuclease activity. Means if DNA dependent DNA polymerase has put some wrong nucleotides, it has not put the nucleotide according to its complementary strand. So here it has an exonuclease activity. It will always read the strand from the 3' end to the 5' and it will always check that are these nucleotides complementary to each other or not. If they aren't complementary to each other, what it will do, it will start cutting these nucleotides. Alright, if there are nucleotides, alright, so it will start cutting these nucleotides if these are wrong placed. Alright, so this activity is known as exonuclease activity, and it has exonuclease activity which will always read the strand from 3' end to the 5' end direction, from the 3' end to the 5' end direction. Alright, now what will happen? Alright, as we know that this is the continuous strand we do have. This is the continuous strand because here we don't have to put primers over and again so that it can be made, but it is a continuous strand because we have just put primer only once. Alright, so the upper strand is known as leading strand. Leading strand. Alright, because we don't have to put here the primers over and again so that it can be made, but it is leading strand because we have to just place primer once. Alright, talking about the second one, it is a lagging strand because we have to just put primer over and again so that it can be made. Alright, so this is the lagging strand. Alright, this we have the lagging strand. So now we have to remove the primers too, because we know that RNA cannot be the part of DNA. Alright, so now we have to remove these RNA pieces. Now what will be happen? Alright, which enzyme will come to remove these pieces? Alright, so now we have a special enzyme which will come and remove these pieces of the RNA so that it can be made. Alright, so now that enzyme is known as DNA polymerase 1. DNA polymerase 1 will be coming and it will remove these RNA primers so that it can be made DNA. Alright, so now we have DNA dependent DNA 1 which will come and start removing these RNA pieces. So these pieces will be removed. Wait. These pieces will be removed. Wait, I have to remove this stand. This stand would be the same one, and it will remove these pieces as well. Alright, this piece will be removed, again this piece will be removed. Now we need something like this is the continuous strand or leading strand, we know that, but here we have to join these fragments, join these Okazaki fragments. So for joining these Okazaki fragments, we do have an enzyme which is DNA ligase, which will come and join these fragments. So we do have another enzyme. What that enzyme is called basically, that is DNA ligase. DNA ligase will come and it will start joining these fragments. Then these fragments will be joined. Wait, let me join it. These will be joined. Alright, so here our DNA will be replicated, like DNA will be made from its complementary DNA. So here the DNA replication process completes. Alright, so how it was made, I am just going to show you people just by the help of that diagram. Alright, so this is the diagram we do have. So let me see here. Alright, if this is the 5' end, alright, and this is the 3' end. Again this is the 3' end because this is the complementary strand of this. Then this we do have 5' end. This is recap of the lecture we have done just now. Alright, now we know that DNA dependent DNA polymerase will come and it has some conditions, like it will always work on the single strand. So we know that this would be the single strand. Alright, but from where it will start, it will always start from the double strand. So here we'll have primase. It will come and put the piece of RNA here. So basically this is the piece of RNA. It has put, like, this is the 3' end, it will put on the 5' end of the new strand. Alright, so here must be the 5' end. So here strand will be made. Alright, here will be the complementary nucleotides of this. Alright, and here we have placed, alright, on the 3' end, and it would be always on the 5' end of the new strand. So basically this is the 5' end. Alright, and it will start making the nucleotides in this direction. So here we have to place 2 or more than 2 primers, which means the pieces of RNA, so that it can be made. Alright, this is the piece of RNA we do have. RNA. Alright, so now we know that RNA cannot be the part of DNA. So an enzyme will come and it will remove these primers, and again another enzyme will come and it will join these fragments, because we know that this is the leading strand. It won't anything to join those fragments. Alright, because here fragments aren't made, but here fragments are made. So basically an enzyme will come, which is the DNA dependent DNA polymerase 1. It will come and it will remove these primers. Alright, these primers will be removed so that it can be joined. So these primers will be removed and enzyme will come and it will join these fragments. Now that enzyme is DNA ligase. DNA ligase will come and it will start joining these fragments so that DNA can be made. So this is how DNA replication happens. So in the next video we'll be starting our next topic. So this is all just about the DNA replication. Thank you.